Hello, my name is Jacob Larsen, and today I will be presenting my research titled The Effect of BMAL1 Knockout and Clock Knockdown in a Mouse Mammary Epithelial Cell Line on the Formation of Alveolar Structures in 3D Cultures. A central goal in our lab is to understand the role of the circadian timing system in lactation, because disruption of this timing system during pregnancy causes a delay in the onset of lactation. A central component of the mammary core circadian clock is the BMAL1 clock transcription factor, which regulates gene expression as shown in Figure 1. Our lab performed ChIP-seq analysis and found that some gene targets of this transcription factor encode for the cell-to-cell adhesion junction proteins E-cadherin and zona occludens 1, which are essential for the formation of milk-producing alveolar structures. A single alveolus is a spherical cluster of cells that is hollowed out in the center, like a basketball. Since the cell-to-cell adhesion molecules are regulated by the BMAL1 clock transcription factor, we hypothesized that if the BMAL1 gene is eliminated or the clock levels are reduced, then the ability of the mammary epithelial cells to form alveolar structures will also be reduced. Our main objective was to develop a 3D culture system approach to study the cell-to-cell interactions among mammary epithelial cells during alveolar formation by optimizing our experimental conditions. Our second objective was to determine the effect of BMAL1 gene deletion or knockout and clock protein reduction on the ability of these cells to form alveolar structures. For this experiment, 3 mouse mammary epithelial cell lines were used and were previously established in our lab. They were wild-type HC11 cells, which acted as the experimental control, CRISPR-Cas BMAL1 knockout cells, and clock-disrupted cells. CRISPR-Cas acts as molecular scissors to cut out the BMAL1 gene and is referred to as the BMAL1 knockout. The clock-disrupted line uses miRNA technology to decrease the abundance of the clock protein and is referred to as the SH clock cell line. For protocol development, we needed to determine optimal plating density, serum and hormone concentrations, number of days in culture, and immunofluorescence staining approaches to visualize the junction proteins and cell nuclei. The lactogenic media was made by supplementing RPMI growth media with serum and lactogenic hormones, which stimulate differentiation of the mammary epithelial cells to form milk-producing alveolar structures. Matrigel was used in a 4-well chamber culture slide to recapitulate the extracellular matrix environment of the breast where alveoli form. To achieve the first objective, we reviewed the literature and worked with experts on campus in a 3D3C facility to develop a 3-dimensional drip culture protocol to stimulate alveolar formation of the mouse mammary epithelial cell line, HC11. The following experimental steps were followed. First, cold Matrigel was used to coat the bottom of the wells of the 4-well chamber plate and provides the bed needed for the cells to form the 3-dimensional spherical structures. Next, the cells were plated at a concentration of 13,000 cells per centimeter squared in a suspension of lactogenic growth media. After the cells attached, a solution of lactogenic growth media and cold Matrigel was added drop by drop, so the final concentration of Matrigel in solution was 5%. This was considered to be day 0 of the experiment. On days 2 and 5, the media was changed and pictures were taken to record the growth of the cells. On day 7, the cultures were stopped by removing the lactogenic growth media and fixing the cells with paraformaldehyde for immunofluorescence imaging using fluorescently tagged antibodies that targeted cell adhesion molecules and cell nuclei. Using this validated approach, we compared alveolar formation in the 3D cultures of 3 separate cell lines. Wild-type HC11 cells were used as the experimental control. BMAL1 knockout and SH clock-disrupted cells were used to determine the effect of elimination or reduction of the respective protein on alveolar formation. We conducted 5 experimental replicates and collected pictures throughout each. However, the experiment was terminated early due to COVID-19 and the quantification of our data was put on hold. The results section of the poster depicts 3 groups of pictures used to analyze the alveolar structures of the 3D cultures. The first 3 show the growth of the HC11 alveoli using light microscopy throughout the 7 days of culture. As shown, the alveolus itself and its lumen expand dramatically throughout the experiment, indicating the strong ability of the HC11 cells to form these spherical structures in culture. The next 4 pictures depict an HC11 alveoli after immunofluorescence imaging. Immunofluorescence imaging allowed for the visualization of the different membrane proteins, with green being E-cadherin, pink staining for ZO1, and blue stain nuclei within each cell. The final 3 pictures show a comparison of the 3 cell lines on day 5 of the experiment. Figure 10 shows that the BMAL1 cells had minimal formation and high amounts of background growth in comparison to the HC11 cells. Finally, the clock-disrupted cells show no alveolar formation at all, which is shown in Figure 11. These cells are similar to Figure 2, however, they never grew past this stage. To conclude, once we are able to complete the next steps of the study, we will quantify our data for alveolar growth and intensity of immunofluorescence staining. Thank you for your time.